Good morning, everyone. I'm Serena, and I welcome you all at our 5th Drug Discovery eConference 2021. Whether you are attending this event for the first time or you have been a part of our conferences before, I hope that your experience at 5th Drug Discovery eConference will be worthwhile. So our conferences have a reputation among the delegates and suppliers as a must-attend event, and I hope that the highly focused business development opportunity through the cutting-edge conference program and the numerous networking opportunities will all contribute to a successful and enjoyable event for you. So before I move forward, I would like to introduce you to our conference platform Hopin and how it works. So as you can see, there are different tabs on the left-hand side, which includes stage, sessions, networking, and expo. All presentations will be executed on the stage. You just need to click on the stage tab and you will enter the room. And then we have a session room, which can only be accessed by the meeting ticket attendees. Then we have a networking room, which will be used for high-level networking with industry professionals. In the break, you all need to go in the networking room and you will have an opportunity to interact with the peers of your industries. So the last room is Expo. And this room will all exhibitions will be carried out in this room. So I hope you all understood the process of Hopin platform. So now I will introduce you to our speaker lineup for today, which includes Eric Halioua. He is working as a CEO and president at PDC Line Pharma. And then we have Antonio Gomez. He is Spain New Venture lead at Johnson & Johnson. And then we have Gregory Macara. He's working as a CEO at Campass Limited. And then we have Robert Soliva. He's working as head of drug discovery at Horizon Genomics. Then we have Dan D. Arider. He's working as chairman of the board at abbio.com. And then we have Eva Mendes. She's working as chief drug discovery officer at OncoHeroes Biosciences. And then we have Golan Bitton. He's working as a CEO at Univo Pharma. And then we have Fred Jacobs, who is working as co-founder and CEO at TYG Oncology. So that's it for the speaker lineup for today's conference. And ladies and gentlemen, our first speaker is Eric Halioua. He will be with us within a few moments. Stay connected. Thank you. Hello, good morning, Eric. How are you?
Fine, good morning. Hello, Sarina.
Yes. Eric, I welcome you at our conference. Thank you so much for being a keynote speaker at our conference. We are honored to have you as our keynote speaker. So thank you so much, Eric. I can see your slides. So shall we start?
Yes.
Okay, so ladies and gentlemen, our first speaker is Eric Halioua. He's working as president and CEO at PDC Line Pharma, and he's going to start now. So, Eric, you can start now, and the stage is all yours.
Okay, thank you, Sarina, and good morning, ladies and gentlemen. I'm going to present you over the next 30 minutes, the activity of PDC Line, which is developing a new class of cancer vaccine based on an off-the-shelf antigen-presenting cell line. So in a nutshell, PDC Line is a spin-off from the French Blood Bank, which is a big organization in France that collects the blood for transfusions and for plasma drugs related to plasma. And they have academic lab associated to the French Blood Bank, and the scientific founder of the company, Joël Plumat, when he was employee and head of the department of the French Blood Bank, has identified in the blood of leukemia patients a very rare leukemia affecting the plasmacytoid dendritic cells. You know, in the blood, you have the dendritic cells that present antigens to the T lymphocyte, and this leukemia was affecting a small proportion of this dendritic, which is the plasmacytoid dendritic cells, that have very interesting property. So based on this cell line, he developed of his population, he developed a GMP cell line that we are still using today as a vaccinal platform to treat patients. So since then, we are a team of 25 people, including seasoned executives from biotech and pharma. In terms of funding, we get close to 32 million euros, 20 in equity and the rest in non-dilutive money. Our cell line has three key advantages. It is a cell line, so it's very easy to expand in culture, and we can produce billion and billion of cells. So the origin of the cells being a plasmacytoid dendritic cells provide very interesting capacity in terms of expansions of antigen, CD8 T cell specific of the antigens, and it is a versatile platform. We just have to load the cell line with peptide representative of the tumor antigens, and we could, in fact, address any kind of tumor, solid or liquid tumor, with our platform. We are a clinical stage company. We have already completed an academic phase 1b feasibility study in melanoma on 9 patients, and we are currently ongoing on a phase 1/2 for non-small cell lung cancer. The pipeline is presented here. So we have 3 key components currently in our pipeline. We have the PDC male, which is completed, and the phase 1/2 is finalized. The result has been published, is not active anymore. We are currently focusing the activity of the company on PDC lung, on non-small cell lung cancer, the phase 1/2, and NEO, PDC neo on neoantigens, which is at preclinical stage currently, or share neoantigens, which are antigens really related to the mutations induced by the tumor. So we signed a first deal in 2019 with LG Chem, the Korean conglomerate. LG Chem is the 10th chemical company in the world, and they have a growing life science divisions, and we license the right for the lung cancer vaccine in Asia. And you see below the total deal value for this deal, $123 million. It was important to validate the platform, and this deal has been signed at preclinical stage for the lung vaccine. One of the assets of the company is definitely the management team. We have people with big experience in vaccine development and cell therapy, like Shanna De Bruyne, that has more than 25 years of experience in vaccine development. She led the registration trial for the MAGE cancer immunotherapeutic of GSK in lung and melanoma. Claude Dédry, the head of the CMC directors, come from GSK as well and have a long experience in cell therapy, where cell and gene therapy, the CMC, is extremely important. The scientific founder, Joël Pluma, who is an immunologist and co-inventor of the technology, joined full-time the company. The CFO, Laurent Lévy, is as well a long experience in oncology. And myself, I'm a serial entrepreneur. I've co-founded several biotechs, sold one of them to Genzyme, and I've been involved in several M&A and IPOs. So we have as well a good board of directors, but I'm not going to spend too much time on that, with very experienced people from GSK, Sanofi, and former AstraZeneca. So the company made significant achievement over the last 24 months. We get the GMP accreditations by the Belgium Federal Agency for the QC and the release activity of product PDC lung. We signed a licensing deal with LG Chem for the development and commercialization of PDC lung cancer vaccine, and the total deal value was 123 million euros. As I said, we get the authorizations to launch the phase 1/2 clinical trials in June for the non-small cell lung cancer. Our medical director arrived in August 2019. In December, we closed a 20 million B round of financing led by Korea Investment Partner, which is a very big, large organizations, 3 billion VC fund that joined the company. We administered the first patients with the cancer vaccine candidate in February. We get the arrival of the new president of the board, Mondher Mahjoubi, which is an oncologist, current CEO of Innate Pharma, in June. And we announced we made a press release to announce the dosing of the first patient with high dose in the cancer vaccine in September. Okay, so few slides now related to the technology, how it works. So as I said earlier, we have with the cell populations of the patients having the PDC leukemia, we develop a cell line. It's an immortalized cells that is stored in liquid nitrogen in bags. And it is a master cell bank that has been approved for clinical use by the French authorities. And from this master cell bank, we are able to develop several working cell bank. And from the back of a working cell bank, we just have to seed a single-use bioreactor. We have 1 liter and 10 liters a single-use bioreactor, and the cells grow in suspensions in a synthetic medium without any calf serum or interleukin. After 10 days of culture, we can produce billion and billion of cells. And we just have at the harvest to split the cells according to the number of peptide we want to load to the cell line, to HLA of the cell line. We load the peptide individually. Then we pull the cells and irradiate to stop the proliferations. I remind you, it's a leukemia cell line. Then we put the populations in a specific container, the technology of aseptic technology with a cryoprotectant, and we store it in liquid nitrogens. And when a patient is ready, we just have to thaw the cells and with a syringe to take the medium with the cells and infuse to the patients. So it's like the COVID vaccine, exactly the same kind of logistic in our case. And the dendritic cells will prime naive T cells in the lymph node of the patients that become memory T cell, expand and go to the tumor microenvironment to kill the tumor, which is the objective of this vaccine. We did several benchmark study to demonstrate the potency of our cell line and the capacity to expand CD8 cytotoxic T cells specific of the antigens loaded on the cell line. And here you see one experiment that has been published where we have loaded the cell line with melanin, it's a specific antigens, and we compare with overall classical approach, which are the conventional dendritic cells or the allogenic dendritic cells differentiated from monocytes from the blood of patients. And we have looked by FACS dextramer the expansions of the antigen-specific CD8 T cells. And as you can see here, we are 20 to 200 for more potent than the classical approach with our technology. So showing you the potency of this cell line. So the manufacturing is extremely easy. The cost of good with the system we have is extremely affordable, and the potency is extremely high. We did as well study with neoantigens and to show that the cell line can expand shared classical antigen or including neoantigens. And the neoantigens are, once again, mutated peptide related to mutations induced in the tumor. And for that, we have identified 2 mutated peptides identified in the blood of melanoma or lung cancer patients published in the literature. And we produce this mutated peptide. You see in red the mutations and the amino acid that has been mutated from the normal peptide. And we loaded with the cell line and put in culture with the blood of 3 healthy donors. You know, the CD8 purified T cells of 3 donors. What you see here, HD01, SD02, and SD03. And you see here the FACS results that measure the extensions of CD8 T cell representative of this neoepitope in red and purple. And as you can see here, after 14 days of culture and 21 days of culture, we have a very strong increase of CD8 T cells that's in the range of 150-fold increase, which are extremely good results, knowing that in healthy donor, the quantity of precursor is extremely low. It's 1 out of 1 million. So it's very, very low proportions of precursors. So today we present the antigens through the HLA-A2 because the patients that provided the cell line was the Caucasians, and in the Caucasian populations, half of the patients are HLA-A2. There is some minority HLA like the B44 or the B7, but we have been able to genetically engineer the cell line to produce additional HLA like the A24 or B35. This result has been published recently, and we know that it works well. So we can expand B35 restricted peptide or A24 restricted peptide, and works very well. So the objective is to enlarge the patient populations and to have a cell line that can address maximum kind of universal cell line or Asian cell line or European cell line to be able to treat effectively each patient. So today we load the cell line with GMP peptide that stick to the HLA and the membrane of the cell line, but we did as well experiments with retroviral transductions of polypeptide of full antigens to have endogenous presentation through the HLA of the peptide. It works well as well. We did as well electroporations of mRNA, and here again we have very good result in terms of presentation. So we have a very versatile platform, and we can adapt the tumor loading strategy according to the indications or the applications. In terms of IP, we have three pillars in our IP strategy. First of it, we have the master cell bank, which is unique, which we have an exclusive license from the French Blood Bank. And the patient is dead now, so it's really impossible to reproduce the same cell population. We have as well 3 patents. 2 has been granted, and 1 is in on the genetic optimizations is in PCT process, and we have as well all the know-how and data related to the manufacturing, the immunomonitoring assay, and the preclinicals. Okay, so let's pass now to show you some preclinical result we have been able to generate with the cell line. You see here an example of humanized mouse. We have humanized a mouse with the PBMC of healthy donor. Healthy donor that were HLA-A2 positive, and then we have vaccinated. We have grafted in this humanized mouse a tumor, a melanoma tumor that was positive for melanA and HLA-A2 positive, and then we have vaccinated this mouse 3 times with the vaccine, which is the cell line loaded with melanA, and a negative control, which is the cell line loaded with an antigens of the flu. And then we have followed the tumor size of the melanoma tumor that were infused under the skin. And as you can see here, after 3, in red you see the infusions of the vaccine. After 3 infusions of the positive control, we completely inhibit the proliferation of the tumor. But if you have the negative control, you see these expansions of the tumor, showing that the vaccine was effective in this model. And what we did, it's at the end of just before sacrificing the animals, we have taken a biopsy in where we have infused the tumor melanoma, and we look for the TIL, identify the TIL, and by fax dextramer measure the TIL that were melanA positive. And as you can see here, they represent 11%. And the TIL are the tumor infiltrating lymphocytes, and they represent 11%, which is a high percentage. So this experiment shows that the vaccine was able to prime and expand T cells in the blood of the mouse that migrate to the tumor microenvironment to make the job, which is to kill the tumor. So this study has been as well published in the literature in PLOS ONE in 2010. Okay, let's pass now to the melanoma trials. We did a study on 9 patients where we have infused 3 doses of vaccine, low dose, mid dose, and high dose, where the vaccine was loaded with 3 different antigens, tyrosinase, GP100 melanA, and MAGE3. And we infused 3 times the vaccine. The result was definitely interesting. We get acceptable tolerability and safety profile. That was the primary objective of this study. We didn't observe any allogenic rejections detected in any of the 9 patients after the 3 infusions. And we have observed as well significant antigen-specific CD8 T cell response directly detectable in the blood of patients, so without any re-stimulation. So showing that the vaccine in vivo in patient was active. And that was interesting because we observe this data despite we infuse just 3 times, which is, we know, not enough. We are going to infuse 6 times in the current clinical trial, and despite we didn't co-administer any checkpoint inhibitor. Okay, and we know that there is synergetic effect with checkpoint inhibitor. And to demonstrate the synergy, we did study with the blood of melanoma patients. Melanoma is a very hot tumor where there is a very high tumor-induced immunosuppressions induced by the checkpoint. And what we did is we have collected the PBMC of these melanoma patients and put in culture with the cell line loaded with melanate once again, and look with the vaccine alone after 1 week of culture, the expansions of the CD8 T cell specific of melanate, and you see they represent we have 15-fold increase, 50-fold of positive cells. If you put the checkpoint inhibitor alone, you multiply by 2 versus baseline the fold, so which is not statistically significant. But if you combine both checkpoint and the vaccine, is where you see this very nice synergetic effect, the 2-fold increase versus the vaccine alone. So showing that we have definitely a synergetic effect between the vaccine and the checkpoint inhibitor. This data has been as well published in 2020. In the next clinical trial, the one that we are currently conducting in the lung, we are co-administrating the vaccine with the checkpoint inhibitor. We did study as well to demonstrate that infusing at more additional injection can boost the activity of the vaccine. And you see here an example of a study we did on nog mouse. In this model, we have humanized with progenitors, the CD34 progenitor. It's cord blood of patient of donor. We have graft versus host disease appearing, not appearing, or appearing a lot later versus the other models I've shown earlier. And in this model, we have been able to infuse 4 times, so which is one more infusions that the other models. And as you see here, we infuse at D0, D7, D14, and D21. And in this model, we have followed in the blood directly the quantity of CD8 T cells that were melanate positive. And as you can see here, after 14 days, and so it's the third infusions, and the third infusions, day 21, we have 1% of all the CD8 T cells that were melanate positive. And after the 4th infusions at day 23, we have 5% of all the CD8 that were positive. So adding 1 boost, you multiply by 5 the quantity of 5.5, the quantity of CD8 T cell in the blood. It's why we have and we are currently in the current clinical trial infusing 6 times, and it's really to boost the quantity of CD8 T cells in the blood of the patients. So you see here the 6 antigens we have selected for the current clinical trial in non-small cell lung cancer. We have MAGE-A4, MAGE-A3, MUC1, New York ESO1, Survivin, and Multimage. And for each of these antigens in the blood of healthy donor, we have shown that we can expand CD8 T cells specific of each of these antigens. It works better for some than the others. It's something that it's normal and we observe in immunology. But we have been able for all of them to show by FACS dextramer the expansions of antigen-specific T cells. The design of the current clinical trial is presented here. We are, in fact, enrolling 64 patients. We are testing 2 doses of vaccine, low dose and high dose. We are measuring the safety, the immunological response, and the clinical response rates, and PFS. And in the first population, it's adjuvant, so just after surgery and adjuvant chemotherapy. And the second population for the two other cohorts is on first-line metastatic patients in association with checkpoint inhibitor. And we have already completed the low dose 6 patients and have enrolling for the high dose monotherapy and the low dose in association with checkpoint. As I said earlier, we are infusing 6 times through IV and subcut the vaccine. So, and just to show you that is our funding strategy, we expect to complete, we raised 20 million euros of funding and to complete the current clinical trials. We expect to get interim reports of the current clinical trial in 22, maybe Q3 or Q4, and to get the final clinical study report mid or Q3 2023, and to have a new round of financing for the second clinical trial, randomized clinical trials in end 2023, beginning of 2024. And on that, I would like to thank you for your attention with a nice slide of part of the team that is based in Belgium. We have a team based in Belgium and France, Grenoble. Thank you very much. Hello?
Hello, thank you so much, Eric, for an insightful presentation, and thank you for your valuable time and joining us for as a keynote speaker for our Drug Discovery e-conference. Thank you so much, Eric.
You're welcome. Thank you.
Thank you. Eric, I will ask the audience if they have any question, they will be asking you on the main stage chat.
Okay.
Thank you so much. Have a good day.
Have a good day.
Okay, so ladies and gentlemen, our next speaker is Antonio Gomez, who is working as Spain lead venture at Johnson and Johnson, and he will be with us. He will be with us within few moments. Stay connected. Thank you.
Hello, that's Sarina.
Hello, good morning, Antonio. How are you?
Fine, fine. How are you doing?
I'm good. Thank you so much. So, Antonia, thank you so much for joining us as a keynote speaker. We are honored to have you as a keynote speaker at our fifth Drug Discovery conference. And ladies and gentlemen, let's welcome Antonio Gomez, who is working as Spain lead venture at Johnson and Johnson. And Antonio, you just need to share your audio and video, and you just need to click on the monitor like icon. Can you see it?
Yes, sure.
Okay, great. Yes.
Okay, my entire screen.
Yes.
Sure. Okay.
Great. I can see it now.
Right.
Perfect.
Okay, so this should be it.
Yes. So, yes, great. Now I can see the full screen. So, Antonio, shall we start now?
Yeah, just one question. So,
Yes.
so we will start at 10:30 or right now?
Yes, actually, the presentation of Eric has been ended before, so you can start it now.
Okay, and will there be time for questions afterwards, or how does it work?
Yes, you will be having that, and people can also ask you questions on the main stage chat.
Okay, so yeah, then it's fine.
Okay, so great. So, Eric, good luck, and stage is all yours. Thank you so much. Good luck.
Okay, thank you very much, Sarina. So, well, first of all, thank you very much to Platinum Global Solutions for allowing me, giving me the opportunity to be here presenting, and yeah, giving some hints, I would say, on how drug discovery based on small molecules, which is sort of the more traditional, let's say, form of drug discovery, is moving forward in our current era, in our current time. So, let me first start by saying a few words about ourselves, about where we come from. So I'm Janssen, but Janssen is part of the Johnson and Johnson family of companies. As you can see in the slides, we are pretty old, 130 years that the company was founded in New Jersey, in the east coast of the US. So at the very least, this means that we are pretty resilient. When you say Johnson and Johnson, everybody tends to think of shampoos and things like that, and this is what you see there. But in fact, the company has three main divisions. One of them is this one, dealing with shampoos and baby creams and baby stuff. But then we have another one, which is dealing with medical devices, and the third one, which is the one that is working on pharmaceuticals. More numbers. So this is a whole amount, a very huge amount of money spent on R&D, which makes sense. It cannot be in any other way, because what we live on is on creating new things, on creating new products. So that means a lot of R&D. And then what you can see below, selling products in more than 175 countries, and more than 130,000 people worldwide, which altogether means that we are very big. If you think on big pharma, we cannot deny that we are really part of big pharma. In fact, we are, I think, I saw some data a couple of days ago. We are the largest company in healthcare, and I'm not sure, but we might be very close to being the first one in pharmaceuticals. So moving forward, Janssen, which is where I come from. Janssen is the division of J&J which is dealing with pharmaceuticals. So of course now we are really very famous. We are every second day we are on the news because of the vaccine. But we have been dealing with pharmaceuticals for many years. You can read through all the text there, but I think the most important parts are what is highlighted in reds. And this is for patients. We have a patient-centric view. And again, this is the only way that we can do it, because at the end of the day, what we want is to create new solutions for the patients. And then the other part is science. We are working on R&D, and what we need to do is to have a very thorough understanding of science, because this is what will allow us to create new drugs, new medical devices, new consumer products for patients. In Janssen, of course, pharmaceutical products. Let's go to more specifics. And yeah, what this funnel tries to show is the way a drug is developed from the start, from the idea up to the very end, which is regulatory approval, and then the drug, the compound in the market. So again, I said that in the previous slide. What we want to do is develop new drugs, and these drugs must be new solution for patients. So where am I coming from? So I am in the drug discovery, and what I'm going to talk to you about today is on the drug discovery part, which, as you can see, deals with a large number of compounds. So in fact, the whole funnel idea is based on the number of compounds that are made on each phase. Why do we do so many compounds? Because our goal, of course, is to find one which is safe enough for first in human testing and which is good enough to be a candidate for clinical trials. In order to do that, we have to make a lot of compounds that have to be tested in vitro, in vivo, to make sure that everything will work properly. So there is a component of amount of compounds. But this, of course, is not all. So it's good that we make a lot of compounds, but it's even better if we make the best compounds. So it doesn't make sense if we just make compounds at random and test compounds at random. It will not work. We need to make many compounds, but ideally we need to make the best, the ones that will give us the more information on how to proceed, on how to move forward, and how to go down this funnel and to end up finding here a compound that is good enough to move into the market, to move into patients. And we are dealing with small molecules. So you have heard before about other modalities of treatments that, they are popping up. But we are still on the good old field of small molecules. This is something that, as I said, is traditional. This is what we have been doing for many years, for many decades. We know how they work. We know how to modulate their pharmacokinetics so that they become drugs in the body. But we have challenges, not only small molecules, but in general, we have several challenges to try to move these molecules to transform what is a small molecule into a drug that can be administered to patients. And I think the biggest challenge is the one that is described in this slide. So just to start and to frame what we are doing. Modern Western medicine is based on a paradigm. The paradigm is there is a correlation between a clinical level, an effect on the clinical level, on the phenotypic level, what you see, the symptoms, a patient, which is, I have pain here, I have this sort of symptoms, and something that is going on deep into our bodies, something that has to do with our biochemical reactions that are taking place inside us. So this has to do with cells and with the biomolecules. This is what you can see in the slide, in the cartoon. So this cartoon is very much what we thought this biological universe of the biomolecules, the ones we have to modify in order to have an effect on the clinical level. By then, this biochemical picture looked pretty simple on relative terms. So what you see there is a neuron. So this is an axon terminal of a neuron. You see the vesicles loaded with neurotransmitters that are released into the synaptic cleft. And then you see the membrane of the second of the postsynaptic cell with a receptor. So the only thing we need to do is to tinker with these mechanisms of neurotransmitters in the synaptic cleft, which are binding there, and then they are removed. So yeah, in fact, this is the mechanism of SSRIs, which, by the way, this is like 1990s, developed in the 1990s. So what SSRIs do is that they block a protein which removes the neurotransmitter serotonin from the synapse and then allow it to work for a longer time, right? So this was the status of science, of our knowledge of science back then. But what's the situation now, and what's the challenge we are dealing with? That's the sheer complexity of the biological systems. If that was not complex enough, this is the picture that we have in mind right now. So you see, it's we have the proteins, GPCRs, bound to the membrane, and then we have all sorts of second messengers going down all the way around there. We have kinases, very similar to each other. We have RNA, which is sending its messages around the cytoplasms. We have DNA, all interacting between themselves and coming up with a very, very complex final structure. What this means also is that what we have always done in science, which is applying a reductionist view, which is, okay, this is very complex, so let's isolate just one of the system, let's say one of the proteins we want to interact with, and that's all. And this is what we were doing. But in fact, the problem is that this is a system where the final properties, they are an emerging property of everything that is interacting. So sometimes, many times, it's not that we can apply this reductionist approach. We need to take everything into account to be able to make sense of this, to be able at the end of the day to modify the biochemicals, the biomolecules, and to be able to have this impact, this correlation into the state of disease, into the symptoms of the disease, or into the disease, the mechanism of the disease as such. So this is very complex, but this is a very big challenge. But on the other hand, this is also very much an opportunity. Why? On the one hand, because as I said before, yeah, I come from small molecules field, but here we find all sorts of other possible ways of intervention into the biochemical processes. We have RNA, as I mentioned before. We can go into the nucleus and we can try to tamper with DNA. That's of course gene technology, and we are seeing more and more examples of how this is going to work for humankind. And we have, of course, the whole cell therapy, CAR-Ts and things like that that can work as drugs. And yeah, that's the first field that I say that this is an opportunity, but then you will see later on the presentation that it's not only that. We see other explanation of why we believe all this complexity can be an opportunity also for us. So let's go more to the specifics. What are the specific challenges that we have in small molecule drug discovery? Well, we need to take into account new targets, new target types. As I said before, so we were very happy dealing with all the GPCRs on the membranes, but we need to look at other proteins, at other ways of interacting to the proteins, and we need to have other types of chemistry that are able to interact with these proteins that were previously considered undruggable. So we knew they were there, but we had no way of finding ligands with affinity for those proteins that could block them or activate them. That's on the biochemical level, so on the first level of this correlation. If we move to the other level, to the clinical level, we are dealing more and more with complex, what I call slippery therapeutic conditions. What do I mean by that? Things like cancer. Cancer, many decades ago, we knew if you have a primary tumor, we knew how to administer one compound and to make it shrink. What happens? Cancer, on a very, let's say, evil way, shows that Darwin was correct, and then it very fast, in many instances, evolve and then create a mutant that can evade this compound that is killing the cancer cells, and then create a whole new clone of cells that are resistant. So that's why it escapes what we are doing with them. Alzheimer, that's another condition. With Alzheimer, we know also for many years the hallmarks of the disease. We've been trying to block the mechanisms that give rise to these hallmarks, the tangles, the A-beta, but it seems that if we block one mechanism, one pathway, there is some redundancy and the disease finds another way to be able to keep on moving forward. That's on the clinical side of things. If we go in the middle, how did we make the connection? We used models, typically in vivo models. And yeah, the fact is that if we rely on those models, they have to recapitulate the human disease. In the examples that I showed before, cancer, yeah, we have lots of mouse models for cancer. If you're a mouse, and you have cancer, it's very likely that there will be a cure for you. And well, luckily, if you're a human, for many cancer, that's true as well. But for others, the cure remains elusive. Why? Because there are sufficient differences between mice and humans to make this not a direct translation. Also with Alzheimer's, or especially even more with neuroscience, yeah, we have conditions that are related to our brain, and our brain is the most different part of our anatomy compared to other species. We can create a model which looks like depression in mice from a biochemical level, but is it really depression? Is it really similar to depression in humans? Alzheimer models, we have transgenics. Those models are really the same as Alzheimer in humans. We don't know, and we have failed many, many times just believing that these models were correct and that what was happening in mice would happen in humans as well, and just at the end of the day, not working out. And then, yeah, part of the solution to all this, which is all the omics technologies that have been popping up and being developed in the latest decades. So we have genomics, we have proteomics, we have transcriptomics, metabolomics, all sorts of omics, which are creating large, gigantic amounts of data. So that's great. But on the other side of things, we need to know what to do with those gigantic amounts of data. We need interpretation, and that's just too much for a human mind. So I think summarizing this slide, I think the problem is that we need now to harvest, to collect the high-hanging fruit. So all the low-hanging fruit, all the diseases that were, let's say, well-behaved, that you could tackle using models, animal models that were reliable, that you could use a reductionist approach for them. All of them have already been dealt with, and we have therapeutic options for them. And it's the most difficult ones, the ones that we now need to deal with. So how do we do that in chemistry, in small molecules? Well, first of all, it's not that chemistry has stopped its development. So organic chemistry, which is what we use to prepare these small molecules, has been evolving. Now we have many, many different methodologies to the ones that we had like 20 years ago. We have new ways, better ways to prepare the compounds. Also, we have new targets that were previously considered undruggable that now are druggable. We have, for instance, things like the protein degraders. So this is something that we didn't think about before. So instead of looking for ligands that have affinity for the protein, we look for things that attach to the protein and then tell the body, from using the internal systems of the body, this protein has to be destroyed because it has to be recycled. And then you have products and things like that. Then we don't have to look very far away for how to improve. So just if we go to the lab next door where our biology, pharmacology guys are working, and they have been applying quite a lot of technology in the past year. So we have high throughput screening, but they have been using 96 well plates, 380 something well plates. So trying to move forward in a much better way. What this means is that it looks like the way forward is not only science, as you can see in the first bullet point, but also technology. So again, if I want to summarize this is about technology, technology, and technology. And yeah, for chemistry, so what we have to do is to join the technology revolution. So technology is all around us. It's not a new concept, applying technology for medicine. So probably if you go to the first, well, not reported, but I would say that probably the first usage of technology in medicine, I can think about Homo habilis in the East African savanna 1 million years ago, just using their newly created stone tools to collect new plants that they could use and that they knew they had an effect on their body. So that's probably the first example of how to use technology in medicine. But now, if we fast forward to the present situation, we all feel that we are living in a world that is really technologically minded. Everything is technology. We only need to go next door again and look at our teenage sons, and they are completely stuck in front of the screen the whole day, probably too much for what we think. But anyway, technology is all around. Technology is solving the problems that we have, and technology is solving problems we didn't know we have. So here you have two examples. So the iPhone and everything Steve Jobs came up with, these are things that he created, and after they were in the market, we couldn't believe that we could have lived before without having them. So how come we could live without an iPhone? How come could we live without internet access? All these sort of things. And then the example below, this is very relevant to the times we are going through right now. So Zoom, Teams, Hopin that we are using right now, all these platforms, they existed before, but it took something very, very specific, which was COVID-19 and confinements everywhere, that everybody started using them. It's not just people in international, multinational companies that needed it to talk with our colleagues in other sites, in other countries, but you saw all of a sudden that grandparents were using Zoom to talk to the grandkids. So technology all over the place. So next logical step, of course, applying technology. And what I'm going to devote the rest of my presentation to is just a couple of examples on how this technology is being applied to the small molecule field. If you remember in the slide where I presented, the one with the funnel, I talked about two main things that we do in preclinical stage. One feature is making a lot of compounds. Second feature is making the best compounds, the ones that will give us, that will provide the more information that will allow us to reach the compound in the clinic and in hopefully in the market as soon as possible. So for the first challenge, how to make more compounds, let's automate what we are doing in the labs. That seems like a logical way forward. And this is not new. For many years, this has been applied to proteins, nucleic acids. So you have a robot such as the one that you see in the top right part of the picture. So what this robot does is that it picks up different amino acids and it links together and it begins building up a peptide, or can be considered, which can be extended also with nucleic acids, just taking different nucleotides and joining them together. So why don't we do just that for the rest of the organic chemistry and creating new compounds? So we don't do that because there are some challenges. So first of all, it's very difficult to generalize. If you think on organic chemistry, on the different ways of creating molecules, there are hundreds, or maybe, well, I wouldn't say thousands, but maybe thousands of different ways or different chemical reactions, different chemical schemes that you have to apply to make molecules combine in the way that you like. So that's one, but it's well, this is what you see there. Sometimes people say chemistry is more an art, like a science, more like an art, because you need to combine in the best way. You need to have hands to be able to make the reactions work. And it's not only that. Every compound, the preparation of every compound is different. It requires different steps, and it requires more than different steps, more different processing. So in fact, it's no surprise that what was automated was the synthesis of proteins and nucleic acids, because all reactions were similar. You had building blocks that were very, very similar, and you just joined them together. But this doesn't mean that we are not going to try. In fact, the first attempt was carried out in the 1990s and was combinatorial chemistry. Combinatorial chemistry was trying to automate. It didn't work out as planned, probably because technology was still not up to the challenge. Also because, well, some of the technologies they came up with, like split and pool, they delivered mixes of compounds that really the industry at the time had problems making sense of. But now we are in a new wave of automation. So now all these technologies are being applied. There are like two general ways. The first one is what I call customized synthesis routes. So basically you use like what you can see in the upper part in this video, and you can see this little robot, which can be programmed to carry out basically in principle any type of reaction. The reality is that most of the time this is used for accessing one compound, and you just automate all the steps of the synthesis of that compound. And then what the robot will do is carry out the synthesis in a repetitive manner. But then you have this generalized platform, which is like a way of getting the best of both worlds, so of organic chemistry, but again, trying to use reactions that are as similar as possible. So what you do by this, in this system, you choose a selected number of reagents, which will allow you to create diversity around a common scaffold. So this scaffold typically comes from high throughput screening, which contains, let's say, the key features for interacting with a specific protein. And then what you want to do in the medicinal chemistry project is to explore around this scaffold and to see how it works. So in order to do that exploration, you use these common building blocks, which you know how they behave, and you can automate the process in a much easier way. By doing that, what do? Well, you carry out this initial exploration, and